So this step here is called the confluence, when cells filled all the surface, the container surface. Confluence denotes the culture stage wherein all the available substrate, the growth area, the container, the petri dish, the flask, is used. And the cells are in close contact with each other. So the confluence هي the phase just before the contact inhibition starts. This may lead to contact inhibition or not إذا عملنا الباساج، إذا نحن جينا شلناهم لهالسيلز وحطيناهم in new petri dish. There are multiple factors that influence the confluence and the cell density and saturation. Cell density بدي اياكم تفرقوا بين شغلتين. Cell density is the number of cells per milliliter of the medium يلي بنعمل له نحن seeding، بنعدهم وعنا تداعي على هالشيء. Saturation density is the density of the cells، يعني number of cells per milliliter square surface area يعني عم نحكي. And this is we have the saturation density in the plateau phase and not before. هيدا كان للadherent cells. For the non-adherent cells cultured in suspension like the hematopoietic stem cells, يعني كل شيء cells cultured from blood, spleen or bone marrow adhere poorly to all the culture dish. So in the body, in vivo, these cells are held in suspension. So we have to culture them in suspension in vitro. يعني ولا مرة عنا blood cells, lymphocyte, platelets يلي هن ولا مرة رح يكونوا adherent on a container surface. Suspension culture are easier to propagate since subculture only requires dilution with the medium. يعني ما بنستعمل enzymes like trypsin حتى نعمل detachment وناخدهم ونعدهم مرة تانية ونرجع نحطهم بالنيو باتري ديش. No, in suspension like dilution of solution ناخد after homogenization we can have 1-2 milliliter, a volume of medium, of culture medium containing cells, and we can re-put or reseed a new container containing fresh medium. بعدين في عنا 3 types of animal cell growth in culture. In simple terms عنا primary cell culture, secondary cell culture, and immortalized cell culture. This is a brief and general description of these features of the primary, secondary, and transformed, and we can have this curve for every culture type of cells. Primary cell culture are the cells explanted directly from a tissue, organ, or a donor organism. Primary cells culture are these cells أو tissue أو organ دغري اخدينهم من الدونر. Example the cell culture of white blood cells, fibroblast مثلا كمان derived from skin, smooth muscle of heart, neurons, etc. يعني بناخد sample of tissue, we can put it in culture, or we can exist in cells, we can have single cells, and we can do the in vitro culture of these cells directly from the organism, from the source. بنسميها primary cell culture. These cells may be capable of 1 or 2 division in culture. أكيد if given the right conditions can survive for some times قلنا من 1 ل 2 maximum 3 days but they do not continue to grow and eventually senescence or die. So معناتها إذا خليناهم هيك. However, and alternatively, in the case of cell that divide in culture, it is possible to passage of subculture them by inducing them to death from the substrate. يعني we can, from the primary culture, we can have some cells, samples of these cells and passage them أو subculture them. أكيد في عنا intervals to respect. So these cell subcultures are or constitute the cell lines, finite cell lines, and every interval time interval يعني كان عندنا each 2 days 4 days يلي حسب الcell types we have to subculture these cells to put them in a continuous culture or subculture. It is possible to develop population of cells that can be passaged and that express stable phenotypes. These are referred to as established أو بنسميهم the continuous cell line. So the continuous cell line independently of time, if they passage control in a control أكيد medium and conditions, we can control the expression of their profile يعني they provide or they preserve the same profile each time وبيكونوا على طول in a expansion log phase, so بنسميهم the continuous cell lines. Some cell lines have arisen spontaneously in normal cells being passaged in culture, but the majority has been obtained by culturing tumor cells, cancers cells, in addition to their infinite lifespan, يعني immortality. Such cell lines frequently share several additional properties that distinguish them from normal cell in cultures. These are the transformed cell lines. So they divide more rapidly. They do not require attachment to a substratum for growth, and they grow in spheroid-like tumor cells. And when reintroduced into animal, they form tumors. يعني they induce cancer formation. Cell lines with these properties are sometimes referred to a transformed cells. So لهلا صرنا شفنا primary culture, primary cell culture direct from the source, limited in time. إذا أخذنا منهم samples وصرنا نعمل لهم subculture in a defined, well-defined conditions and controlled rate of proliferation, we constitute the cell line. These cell lines is maintained infinitely in these conditions على طول مع subculture, we constitute the continuous cell line. If they have the ability to induce cancer formation, so صار في عنا mutations that induce a transformation from normal cell line to a cancer cell line نسميهم transformed cell lines. When لا الكونتينيوس ولا الترانسفورميشن pathway منفوت فيهم, so these cell lines will undergo senescence and death بعد ما نكون وقفنا السبكالتشر تبعتهم, which is mean انه صار في عنا contact inhibition and cell death. Here some advantages and the limitations of the utilization of primary cultures. The capacity of biotransformation and the tissue-specific functions are good points, are positive points. For the capacity of biotransformation, in many cases, the metabolic profile generated by a primary cell culture, مثلا hepatocytes, إذا أخذناهم عندهم greater similarity to in vivo than does the pattern seen with subcellular functions used as exogenous source of biotransformation. يعني these capacity of biotransformation عند الاباتوسايتس in vivo عندهم behavior معين معودين عليه. When cultured in vitro they can conserve this capacity of biotransformation and they will act like in vivo which is very good to extract the metabolic products and use of these products in future applications. The second advantage is of primary culture is the retention of tissue-specific functions. So, for example, primary culture of myocardial cells, مثلا, consisting of synchronously beating cells in vivo. When cultured in vitro, these cultures, these cells, if they exposed to a cardiotoxic molecules بده يصير في عنا arrhythmia and cessation of beating were observed. So we can نعمل screening of molecules to a type of cells and we can assess this in vitro and not in vivo. مشان هيك when culturing مثلا myocardial cells they will conserve this beating in vitro. يعني بنشوف بالباتري ديش myocardial cells عم يعملوا beating like in vivo. We can test a molecule تنشوف شو تأثير عليها مثلا يا بتوقف البيتينج يا بتسرع البيتينج etc. For the negative points or limitations, cell isolation for each experiment عندهم a very heavy protocol وكتير صعبة. Limited lifespan and change لأنه مثل ما قلت لكم primary cell culture we can maintain them in vitro من تقريبا maximum ل 3-4 days منو أكتر. For the continuous clonal cell lines أكيد more resistant to toxic insult. ليه؟ لأنه عندهم usually they freshly isolated short-term cultures and we can maintain them continuously in أكيد and under well-controlled conditions. Some cell lines maintain or secrete tissue-specific proteins and enzymes which can be used for examination of as biological markers, which is very good. ويمكن هذا aim principle one to culture and to continuously culture cells. Limitation homogeneity of cell type لأنه many cell lines have been available for extended time periods having undergo many in vitro passages. So كل هيدا stress للcell. So before use cultures should be carefully examined of homogeneity of cell types لحتى نشوف هل أنا عندي 100% the same cell type بعد هالcontinuous passages بعد هالcontinuous و subcultures ممكن منهم يصيروا transformed مثلا ومنهم يبقوا مثل ما هن. So الhomogeneity هون كتير مهمة. Cell line must be clean of contaminating mycoplasma, bacteria, viruses, and sterile techniques must be strictly adherent to لأنه contaminating هيدا على طول الكشمار تبع every culture وأكيد إذا dedicated to a implantation in vitro عند الهيومن لازم تكون أكيد mycoplasma, bacteria and viruses free. So لازم الكنترول البروتوكول of control of contamination يكونوا very strict and very يعني بده يكونوا continuous كمان مع السيل لاين. كل ما ضلت السيل in vitro أكتر وقت كل ما تعرضنا أكتر لcontamination. So this is the third part of this chapter. I will upload a video number 1 رح يكون اسمه for the protocol of culture of animal cell culture and animal cell passage of adherent and suspension cells. بيكون مصور عنكم بقلب لاب أحسن ما نقعد نشرحه and you have the step. كمان حطيت لكم الsteps على سلايد تيكون موجود قدامكم with explanation. Voilà, هي كانت this part for the week number 4. We will meet in a new Zoom session. من هلا لوقتها stay safe, stay home. Bye.